Good morning, and thank you to be with us here. Thank you for the speaker also. This is a lecture that was coordinated with the help of the Christiana Care, especially with our chair of the external advisor of the Alliance program, that is the one that are patrocinated this lecture. Dr. Khan is with us here. I think that Dr. Khan is with a limit time. I prefer that you introduce yourself, give us a small words before we begin with the introduction, Dr. Khan.
Buenos días a todos. Gracias por estar aquí. Ojalá estuviera en Puerto Rico contigo. En nombre del CTR del Delaware, gracias. Good morning, everyone, and thank you to Dr. Mayor for putting this together today. Thank you very much on behalf of the Delaware CTR, which is one of the collaborating institutions for Puerto Rico. I want to thank the principal investigators for the Puerto Rico CTR, the Alliance, Dr. Carlos Luciano, Dr. Ivana Lifta Moru, and all the site coordinators and leaders, including Dr. Angel Mayor, and my other colleagues, including Dr. Valerie Woina, who's here today as well. We are so pleased to be collaborating with you. The whole idea of the idea states across the United States and territories is to share lessons learned. We have learned a lot from our colleagues in Puerto Rico around community engagement and community engaged research. We share best practices, we share ideas, and it is my honor and privilege to be participating with the External Advisory Committee for the Alliance CTR and to have hosted colleagues from Puerto Rico CTR to our Delaware annual meetings. You have a wonderful group of colleagues today, and while I won't be able to stay for the entire meeting, I will be coming to see you all for the scientific retreat in about a month. Dr. Mayor, again, thank you so much. Mr. Derek Bowden, Debbie Moore, Dr. Brian Levine are some of our best colleagues in Delaware, so we're sharing with you our best practices, and I'm so grateful to Derek for having taken the time especially, and to Debbie and to Brian for their time. So, Angel, thank you again, my friend, and to all of my colleagues there. Hope to see you very soon, and I hope you have a very successful workshop today. Muchas gracias.
Dr. Khan, thank you for your words, and thank you for this Spanish word. We know that Spanish is not your first, you're one of the second language, but was very good, excellent. And I want to say you that we have here a very young group of persons that I am very happy. I think that we have very new investigators, so it's really nice that we have this new person with us that will be the future of the research in Puerto Rico. So I think that we will have advantage of all the things that we are presenting here. Thank you, Dr. Khan, again. So right now I begin with the introduction of our speakers. As Davis mentioned, we have three speakers. One will be present, that is Mr. Derek Bowden, and the other two will be via Zoom. So, but Mr. Bowden will be coordinate all the changes on all the elements. We have person in person and person also in Zoom. You can make your questions when they are, if they want to, you can raise your hand and make the question so that Mr. Bowden could ask your questions. And there are things that this is record, so but you have to be aware that we are recording this conference. So beginning here, I will introduce you Derek Bowden. Derek Bowden is the IRB director of the Christiana Care. As they mentioned, is one of the largest academic centers in northern Delaware. Dr. Derek managed all the aspects of human research project program in the Christiana Care, include the IRB, research compliance, education, support clinical research activity across many departments, including cardiovascular, maternal fetal, infection diseases, nursing, and also the residence and fellowships program of Christiana Care. She was also director of the research protection at Mainline Health at Philadelphia. He was also provide regulatory oversight of the Lankini Institute of Medical Research. And he was also involved in or worked with the health department in research that involved human subjects. The other person that will be with us is Ms. Debbie Moore. Debbie Moore is the director of the research in the Christiana Care. She oversees the grants and clinical pharmaceutical tracks for the Department of Medicine in the Christiana Care and include endocrinology, infectious disease, neurology, pulmonary, as well as cancer and health, vascular care, cardiovascular trials. Also, the Helen F. Graham Cancer Center and Research Institutes, the NCORP, grants and the pharmaceutical trials and the Christiana Care Emergency Medical Research Department. She is involved in, have been involved or have big background in phase 1 and 2 pharmaceutical trials and grants. And since he began, initially he began as a clinical coordinator, and after that they changed to supervisor management and director of this trial. She is involved also in contracting contractors, budget submission, progress, resources allocation, study evaluation, and the closure. She has a degree in nursing of the Nursing for Delaware Techniques. The other person that will be with us is Dr. Brian Levine. He is the Associate Chief Academic Official and the Design Institutional Officer of the Christiana Care. He oversees the graduated and undergraduate medical education, as well as the virtual education and the simulation training certain, continuing medical education, the medical library, and also the allied health programs. He has an extensive background in graduate medical education, having served as a program director of the Emergency Medical Residence Program since 2012, and as associate program director from 2006 to 2012. He's a clinical professor of the emergency medical at the Sidney Kinnell Medical College at the Thomas Jefferson University. Dr. Levine his medical degree from the University of Vermont and is graduated of the Emergency Medical Residence Programs in Christiana Care. He is also served as the Associated Medical Director of the LifeNet Aeromedical Transport Program. So now I will be leaving you with Mr. Derek, but we began with the letter.
Great, thank you. Thank you, Dr. Mayor, for that introduction. And as you hear, I have a great esteemed group of colleagues that I work with, and I don't know how I got so lucky to be the one to actually come here on site. This is wonderful. And Dr. Mayor has been a wonderful host. Thank you to Nighty and Nicole and Luis for assisting us in getting everything set up. I'm already made myself at home, but I have to leave tomorrow, unfortunately. I wish I was here in a week. Because it's supposed to be nice on Saturday, right? So, of course, I'm leaving the day before. It's going to be beautiful weather. So just a couple of introductory things, and then I'm going to actually turn it over to Debbie Moore, who's already on, and then Dr. Levine will be joining us a little later, probably at the 11 o'clock hour. You'll see the slide deck here. It's 88 slides. This is overly ambitious. I understand. There's no way we're going to get through all this. I'd prefer if throughout, if you have questions, raise your hand, interrupt me. Those online, you can drop something in the chat, and the folks in the back will alert me to those questions. I'd rather us focus on the things that you need. We'll skip over some of the slides, but the slides will be made available to you, following the presentation after the evaluation. I've embedded some links in there, things to think about, things to look into further. There's just a lot, right? Clinical trial designs and human subjects implications. It's a semester-long course to discuss it. We're trying to cram it in 2 hours. So we'll get through what we can, but please stop me if there's something in particular you want to focus in on. Just a little bit about Christiana Care, really quickly. So we're a large health system, huge volume, but our research footprint is fairly small considering the size of the health system. But because the health system is so large and the volumes are so high, we actually do have a quite sizable portfolio. Have over 500, close to 600 active protocols. And that keeps us busy. We're a fairly small research administration team, as you'll see on this slide. This is kind of the structure of our research administration. So we all report up to Dr. Omar Khan, that you just heard from. He's our Chief Scientific Officer. I run the Human Research Protection Office. My colleague Debbie runs the Office of Research Coordination. She's the Director of Research. And then we also have a sponsored programs office to manage like grant submissions and things, and then a Office of Research Finance. Most recently, a department's called iReach. It's the Institute for Research, Equity, and Community Health that was recently added to our research administration infrastructure. So we're a great, really close-knit team, work together frequently, oftentimes multiple times a day. And that is really essential as you start to develop a more and more robust research program. The investigators know the science. The investigators know the medicine. It's the research administration that needs to support that research, right? We're there to facilitate the research. You may read, you have to comply with all of these regulatory requirements, and it's overwhelming. You know, my philosophy is that we want the investigators to be focused in on the science and the medicine, and we'll focus in on all of those rules that you have to follow. But it's obviously a luxury, right, to have a great research administration team. My office, the Human Research Protection Office, we have 4 staff, so we're small. Myself, an IRB coordinator, an education specialist, compliance administrator, and then the coordinator also serves as an administrative assistant. We used to be just an IRB office, right? That was sort of the primary focus, was the function of the IRB, and then we expanded it out to the Human Research Protection Office because there are a lot of activities like nursing research, maybe some like qualitative research. Some residents will do like chart review type things. These things may not need to go to the IRB. If you're doing some even specimen research, as you've seen on the NIH human subjects clinical trial forms, where it says, well, are you using human specimens? And you may not require IRB review. And that confuses a lot of people. So we support all of those activities. A little bit about our most recent fiscal year, just the volumes and the type of reviews that we do. We have currently closer to 600 active protocols across the system, mainly oncology. That's our big, research division. Also maternal fetal medicine, Department of Medicine, Family Medicine, cardiology, and then many other areas that we support. The bulk of our reviews by the full committee, we have 2 IRBs, 2 committees. We reviewed last year 108 full committee reviews, full protocols, also 145 expedited reviews. And we do a lot of studies with external IRBs. You're seeing a lot more work in the single IRB requirement, especially if you're NIH funded, you're multi-site. You can't each review your own research. You have to rely on a single IRB. So we spend a lot of time negotiating those agreements. They're called reliance agreements. Determine who's going to serve as that IRB. And then down here, we also support a lot of quality improvement, performance improvement, not human subjects research project, right? All those activities that sort of fall in that gray area. Is it research? Is it not research? Is it human subjects? Isn't it? We don't want to leave those project leaders on their own. We still want to support those activities as well. I spoke to this group, I think, back in March, and didn't know I would be here this quick, but this is great. So I always include this slide in every presentation I give, because whenever someone comes to me or calls me and asks, what rules apply? Like, what do I need to consider? My first question is, who's paying for it? It all depends on where that dollar is coming from. I told Dr. Mayor last night, the government doesn't give you money for free. If the federal government's giving you money, you're going to have a lot of work to do to spend that money throughout the life of that project. So depending on what agency is supporting that activity, those rules will apply. So this slide, it just represents, the different agencies, and each group sort of has different rules, and these rules don't always align. That's the thing that's really confusing for most folks. And, when investigators come to me and say these two regulatory rules contradict each other. I can't understand this. It's too complicated. I say, well, it's good for me because I wouldn't have a job if it were easy to navigate. So, you've heard of the common rule agencies, the 45 CFR 46. That's our main code of federal regulations that protect human subjects in research. There are 21 or 22 agencies. I've listed the ones here that fund research. A lot of agencies don't fund human subjects research, so they're not applicable. The Department of Defense is a big one. But also NSF and NASA, Department of Justice. And then on the right, you see NIH. It's technically a common rule agency because it's part of the HHS, but it has its own category because the amount of research that the NIH funds eclipses all other agencies globally. In fact, if you put all agencies across the globe that fund human subjects research in one pot, the NIH is still larger. So that is the bulk of the funding, and it fluctuates from year to year. It doesn't mean it's easy to get, but because they fund so much human subjects research, their rules are the most complex by far. The one in the center here is the FDA. The FDA does not fund research, and they also don't conduct research, but they are the ones that approve research that leads to the approval of investigational products, devices, drugs, biologics. So they have an extensive list of criteria as well that are very complex. And again, sometimes the FDA and the common rules do not coincide. So we'll always look at it and determine which rule provides the most stringent protection of human subjects, and that's what we'll defer to. But then again, in some cases, the FDA won't allow us to defer to any other rules. We must comply with their rules. So yeah, it can be a nightmare. So our basic ethical principles are based on three. This is really how the federal regulations were codified out of these three basic principles, what we call the Belmont Report: respect for persons, justice, and beneficence. And respect for persons can be translated into the informed consent process for study participation. Informed consent is not a document. It's a process. It should be an ongoing dialogue. Once a participant enrolls and signs that document and you retain that document, that informed consent process really should continue throughout their active participation. Justice is the requirement of equitable distribution of research burdens and benefits. So we all share in the burdens and benefits of research. So it's balancing that level of risk. What level of risk is tolerable in order for us to hopefully advance science, advance medicine, so that it benefits, future populations. And beneficence, the requirement that the risk to human subjects is justified by the value of the knowledge that the study is expected to generate. So now I'm going to turn it over to Debbie Moore, who hopefully is still there, patiently waiting. Debbie has decades of experience being a research coordinator. I think she started when she was 5. Isn't that right, Debbie? So over 40 years that she's been doing this. So she's really an expert in effectively managing clinical trials. She's going to give us some information pointers on developing budgets and managing those nuts and bolts of those trial programs. And Debbie, you can go ahead and take over, and I'm going to advance the slides as you speak.
Okay, sounds good. Good morning, everyone. Thank you for inviting us to participate this morning and be able to share a little bit of what we do up this way. I do have one slight disclosure. I'm a little jealous of Derek because he is down there, although we shouldn't complain. Our weather up here this past week has been pretty nice for us for November, so can't complain. All right, so I'm hoping I can see this well because like Derek's, the pictures are kind of, let me move this out of the way. Okay, there we go. So what I'm going to talk about this morning is a little bit about how we manage the CTR trials. If we were to go into the other things that I do, which is the pharmaceutical clinical trial coordination, it's a little different animal. So I'm just going to stay focused on this today. But if anybody has any questions about anything else, I'll try and answer them. So what does a research coordinator need to know when they're submitting or helping their PI submit for funding? The biggest thing is to start early. Give yourself enough time to get organized, figure out who's going to do what, and don't wait to the last minute. it's not going to set you up for success if you're scrambling at the last minute and not submitting what you need to on time and in the right way. Another important thing to do is to identify the appropriate funding opportunity for the research project. So you've got your idea together, you kind of have your protocol roughed out, and now you're looking for the money to help or funding to help support that work. And so on the CTR site, which sounds like some of you are already familiar with, there are different types of funding opportunities there. So you need to kind of read through it and pick out which one meets your needs and which one you actually qualify for. The other thing is once you determine that, you want to look and determine what forms are needed and the timelines for those submissions. I usually tend to just print everything out or save it electronically so that those are kind of my working copies in anticipation of the final to submit when we're all ready to go. It's really important to follow the guidelines for your form completion, and that includes, how many pages are needed for each section, what are the formatting requirements, because if you submit and you haven't followed the rules, it can delay or get your project kicked back. The other big thing is the PI, principal investigator, must register as an Excel user because they are the ones that are going to be responsible for uploading the forms. Once you kind of know what funding you're going to go for, you need to develop a budget. And this is a big chunk of it. You need to identify who your personnel resources are going to be. Who's going to do the work? You think of it that way. You're going to have a principal investigator. You're going to need somebody to research nurse, research coordinator to help kind of keep everything together, maybe potentially help run the project to a degree, research assistant, data analyst, anybody that's going to work on this project and have some significant effort or time to the project. You need to think about what is that going to look like? And what are they going to do? Are they going to do procedures? Are they going to do testing, surveys, data review, collection, focus groups? What is the work that's going to get done? And when are they going to do it? So if you're looking at a 2-year funded project, it's always helpful to look and see when this work is going to be done. Is it going to bulk of the work going to be done in the beginning, in the middle, or the end of the funding period? Typically, as much as we would like to start on time, we don't. So if you think of it, kind of keep in the back of your head, okay, I'd like to give, the research coordinator more effort in that first year than the second year, but then you're starting late. So maybe some of the bulk of that work is now going to be pushed to that second year of the project. So you got to kind of think about how is this going to lay out, and how do you need to kind of stretch out the effort that's going to be done over the time period, and how long is it going to take them to do it? So once you have your project, how many hours are going to be needed to complete the work for that time frame? And then you got to think about the non-personnel expenses. People tend to forget the little odds and ends, and they can add up if you have forgotten to add them in. Simple things like exam gloves, blood draw tubes, paper, pens, anything that you're going to need to support that project. What about equipment? Do you need to buy equipment, supplies for the equipment, disposables? You need to think about, getting quotes for, pieces of equipment. Are you going to lease it? Are you going to buy it? Those kinds of things. It's kind of all that kind of gets rolled into that budget process too. And then there's travel. The big thing with the CTR is that you are expected to present your project once it's completed at the annual meeting. And you're allowed to include those travel costs in your budget. So you want to keep that in mind too. Like, where is that meeting going to be? And, what kind of, meals, all those kind of things get rolled into that travel budget. And then there's the big other category. You've got publishing fees, consulting fees, transcription fees if you're doing focus groups and you're recording and you're going to need it translated, patient stipend, patient travel, patient meals and snacks. So always remember, we must, because this is federal money for the CTR, we must follow the NIH policy for allowable and non-allowable expenses. I've put the link in here to that to that page in the NIH policy if you want to save it, or I'm sure the slides you're going to give it are going to be provided after the talk. But that's the link where you can go and look if you have any questions. And if you don't know, always ask. You can always call someone and ask. The big other thing is to calculate the personnel cost. And so you want to consider how many hours of time and is going to be given per person, and you want to change that into a % effort, because on your budget justification form, you're going to need to put in the calendar months. How many calendar months? And that's part of the budget justification. So I put a link in here also that it's also on the NIH site that can convert your time into the calendar months. It's just easy, so you don't have to do hard math. It'll do it for you. So you're taking the hourly rate times the % of effort, which is going to translate into the salary that you're going to request. And the salary requested you're going to add your fringe in there, and that's going to give your total funding requested for that person. You need to calculate the cost of supplies, the equipment, travel, publishing, and other. And then you're going to take all those total direct costs, and you're going to add, and I'm assuming you guys have your hospital rate agreement for F&A like we have up here. And I'm assuming you have to kind of follow that too. I don't know. So I just kind of threw that in there because I wasn't sure. And so whatever that negotiated rate is for to do research, that would be the rate that you would need to then add into that. Another thing to note, the CTR grants do not fund PI salaries. And the cost of that PI salary is going to be assumed as cost share to the institution. In other words, the institution is going to support your work that you're doing on the project. You're not allowed to apply for any funding as the PI for the project. And then the % effort is entered on the budget justification form, like I said, as calendar months. And then you would also write in there no salary requested for the PI. And any personnel whose base salary is over the NIH cap, and that usually changes yearly. So you would need to look on the NIH site to find out what that number is. That also becomes cost share to the institution, and that becomes a calculation. So I'm trying to give you like a really big umbrella view of this instead of getting too much in the weeds of how to actually do all this, which, if you have questions, we can, follow up later, but just trying to give you an umbrella view. So the forms, so you want to, like I said, you can either download all the forms or you save them electronically that you're going to need to submit for your project. And you want to complete the forms per instructions. They do have instructions there on what you need to do and what needs to go in there. If in doubt of what is required to populate a form, always reach out to the contact, because there should be somebody listed for that funding opportunity on the Excel CTR website. And you can always reach out to them if you have any questions. And I do recommend that you do that, because I still do that all the time. If we have something comes up, there's always something that's not like it was before, and you have to ask. It's always good to ask. Like I said, I mentioned earlier, incomplete forms or incorrectly completed forms may result in your project not being reviewed or funded. So that you don't want to get to that point. You put all this work in, you want to have a good submission. And then the PI will need to upload all the forms once they're completed. If you can submit before the submission deadline, I would recommend doing that, because that way you may get some feedback, and then you can make any corrections or changes before the actual deadline. You'll receive an email that your project has been funded, and that's a big yay. So now you're ready to get moving forward. You've been funded. Now what? So if you've not already submitted your project to your institution review board for review and approval, you'll need to do it now before any work can begin on your project. And then once you have IRB and funding approval, it's always good to schedule a startup meeting with your team to go over the project, because some time has gone by. Everybody's been kind of working on their piece of getting this submission done. So now it's time to regroup and discuss role responsibility, tracking of hours, because you want to track, everybody's effort, their hours to the project, task delegation, because it's important that no one involved in the project will exceed 100% of their contracted time to the institution, because, in theory, we all know we work a few extra hours every now and then, but from however your status is at your institution, whether you're a full-time employee or a part-time employee, you have a set amount of hours that you work for that institution. And so that becomes 100%. And so you're not able to work over that amount of time. And this is With federal money, they want to make sure that people aren't going over the 100% effort. The other thing you want to do too now is to purchase your supplies and equipment that you included in the budget. You want to save all invoices and receipts for documentation, because in the event of an audit and to track your spend on your projects, you're going to want to have documentation for that. I would suggest maintaining a binder for all the project related documents that are needed, and the ones that you aren't able to scan in or save electronically. I would, put them all in a binder so they're all in one place. You're not searching for stuff as towards the end of the study and somebody says, hey, what did you spend on that? I don't have any record of that. Or where's that invoice? It's always good to kind of keep everything in one spot. Any equipment that you purchase may need to be cleared by your clinical engineering department. Is it going to, if you're plugging it in somewhere, is it going to blow a fuse? Is it going to be too much load on the system? All these little weird, goofy things that sometimes you forget about because you wouldn't want to get ready to go with your project and you've purchased this piece of equipment and now you can't use this piece of equipment because it's not going to work, where you want to plug it in. And any project supplies or equipment can only be used for the funded project. Once the project's done, then you can follow up and see if you're able to keep the piece of equipment, and then can it be used for other things. And so in that instance, if you're allowed to do that, to use it for the next project that's going to be very similar, say you're building, your next project is building off of your current one, and you're going to want to use the same piece of equipment, you just have to let everybody know that you're doing that, and that you can't include that in your next budget to that you're going to purchase this piece of equipment again because you already have it. If you are enrolling human subjects, you'll need to register your study on clinicaltrials.gov. You can navigate to the study record manager's link for more information. And I don't know, Derek, is that kind of falling off the screen, or is that link visible?
Yeah, we can see it. Yeah, and it'll be in the slides.
Okay. Okay, yeah, I think it's just a ribbon at the bottom of mine. I can't see it. So that is for clinicaltrials.gov, and that will give you more information there. So if you're doing a clinical trial with human subjects, and if you're enrolling subjects, you want to have a recruitment plan. Discuss with the team who is responsible for recruiting the patients and how you're going to recruit these patients. Are you going to have go through referrals, ads, chart review? Any patient-facing materials need to be reviewed and approved by your IRB. Create source documents for data collection so that you're collecting the same data across the board on all patients. You want to maintain a database of all screened and enrolled patients so that if for some reason someone has a question about something, you can say, oh no, that patient didn't qualify, we weren't able to enroll them, we screened them, we weren't able to enroll them, and this is why. So that it's showing that you were, part of that reason is to show that it just wasn't because you didn't like the looks of them and you didn't want to enroll them. That has to be, has to meet the inclusion exclusion criteria for, for maybe being a screen failure for getting that patient in. And then very, very important, that's why I bolded it, before any study related activity is done, the patient must sign the informed consent form and you must give them a copy. And then also make sure that the patient understands what they are consenting to, the timelines and the follow-up, because you don't want to enroll somebody in a study and they, it looks good. And they're like, especially if you're providing a stipend for their time to participate in the study, and they're just kind of focused on that. And then all of a sudden, you're a month or two in and they're like, well, I don't want to do this anymore. I didn't know it was going to take this long. Or did I say I was going to do that? So you want to make sure they have a very good understanding of what they're signing up for and what they're consenting to. And, if they have any questions. So the other thing is you only want to perform IRB approved protocol related activity, and collect data on source documents and keep the patient study chart. We like to keep the old fashioned paper charts because sometimes it's just easier to have everything in one place for when the patient comes in, but you can do it electronically too. But you just want to make sure that you're saving all of that, whether it's paper or electronic, and that it's in a secure location. And that only those people who are involved with this study have access to it, because you have to worry about HIPAA. So you want to document in a clinic note your study visits. You want to say if there's any changes in health or meds or adverse events. Follow your IRB policy for notification of serious adverse events or unanticipated events, deviations, or violations of the study protocol, because that's very important. If you're conducting focus groups and recording sessions, check with your IRB, your institution legal policy for any additional consent that may be needed. Here we have an additional consent that we provide to patients participating, especially if they're going to be filmed. That we have our legal team has a separate thing where they just have to kind of initial and sign. If you're providing a stipend, meals or snacks for study participation, being sure to maintain records of payment. So same thing, like I said, anything that you're doing with purchasing equipment, anything that requires an invoice or tracking of the money, keep track of that. Let's see. Submit any interim reports as required by the funding opportunity and or your IRB. So if a continuing review is going to be done because it's a 2-year study or study that we did, we started, I guess it was last year, Derek, that the IRB requested that within 6 months of us starting the study, they wanted us to provide an update 6 months in instead of waiting that full year, just because of the type of study it was. So follow your IRB guidelines on when it's expected for you to submit any reports, and then also with clinicaltrials.gov and with the funding opportunity, they'll also let you know if you have to provide any type of annual reporting to them. All right, so study's done. Now what? So all study activity must be completed, including the data analysis, by the end of the funding period. You cannot invoice for work done after the date of the funding period. You can check with the CTR coordinating center if there's a grace period to submit any outstanding invoices for expenses that occurred in like the last few weeks of the project. And that typically happens because you still have, you still have people working on the project up until the last date or the end of the funding period. And so there usually typically is a grace period to get those invoices in so that you can get paid. And a biggie is it is easier to manage the spend of on the project as you go along and not wait until the end to submit invoices. You don't want to find out that you've overspent your funding and don't have funding to cover your costs, because then what happens is that your institution is on the hook for those costs and they might not be very happy with you that you have done that. So you have to kind of be a good steward of your financial reconciliation as you go. It's just easier. We do it on a monthly basis with like with salaries and that type of thing. It's just easier to keep track of, and we kind of do a spend down. So we have a spreadsheet where we say, okay, this is how much money we have for the PI. This is how much we have for the research coordinator to start with. And then as the month goes along, we just track it down to the point at which, here's how much you have left to spend. You have 2 weeks left on the study. You've got like 3 hours of time left now. So figure out how you're going to make that work. So we don't, so we stay on track for spending.
Okay, thank you, Debbie.
Thank you, guys.
Hey, Brian, are you there?
Yes, I am.
So I'm going to go ahead and turn it over to you. You're the next slide anyway. So thanks for jumping on a little bit early.
My pleasure. And you're going to.
And they've already been introduced to you. So we already read your little bio.
Oh, great.
Or long bio.
Are you advancing the slides as well?
I am. Yeah, I have the slides all here and I will advance them as you speak.
Perfect. So it's a pleasure to be here. Thank you for this invitation. And as Derek mentioned, I am an emergency physician by training, but I wear a bunch of hats, and one of which is the chair of the IRB at Christiana. And I also have done a fair amount of research work and try to get out one manuscript every few years. So I've been playing a role in a variety of different levels, from principal investigator to presentations and manuscript production, all the way now to chair of the IRB. So what we're going to do for a few minutes is discuss the importance of being a principal investigator. So there are commitments that are really important for us to recognize, because the PI, the buck stops there. The principal investigator really has to assume the responsibility for the conduct of the entire study. And that's a powerful position, but one which should not be taken lightly. And I'm going to discuss some errors or some pitfalls of where principal investigators sort of fall short, and then the trial could fall short and collapse because because of that. So we've seen these, and in our experience, I'll try to give as much examples from what I've seen in my experience, but by following some simple rules, the principal investigator can really be the glue that holds a study together. So they really need to be familiar with every part of the trial itself. They need to know if there's a product, they have to be familiar with that and its usage. They have to comply with good clinical practice and all its regulations and be prepared for all documentation, even though he or she relies on those around them for administrative purposes. The PI still needs to be aware and understand where everything is. And then maintaining a log can help with a lot of the organization. Now, there are definite research managers and others that will help you as a PI, but it's still ultimately your responsibility. And a lot of PIs say, well, I didn't know or I wasn't sure. It doesn't matter. Going in, the PI needs to know that it is their responsibility regardless. Now, there are resource utilization issues that always come up. And what I see, especially in a training environment, is an inappropriate research question. And the PI usually not having enough experience or knowledge to recognize that they're biting off more than they can chew, or that this research question is going to take 12 years to recruit the numbers you need. So hopefully their PI has the wherewithal to present appropriately and have a knowledge of research whereby these projects don't flounder or it's going to be too difficult for you to recruit. So the PI needs to look at the ability to recruit the appropriate numbers and on time. That's important. And this really is difficult. And things that I've seen over the years take 20 to 50% longer than you anticipate. And that's a classic mistake that PIs make, is they don't realize that things don't happen immediately in research studies. They're complicated. So you need to have appropriate support staff as well who are qualified, who understand what the processes are, and you need an adequate place. Usually hospitals, but there's many outpatient settings and a variety of settings in general. We've had some of our residents go to foreign countries and perform research and bring that research back. So you really have to know your environment, and the staff that you have should really understand the protocol as well as you, if not better, because they're going to sometimes be performing a lot of logistical activities. So they need to understand the study responsibilities as well. The medical care is really important. Our goal, obviously, and the IRB's goal, is to protect the patients, but the PI has to assume that responsibility as well and ensure that all decisions that are made in a trial that involve medical decision-making are done by a person who's qualified, usually a physician who's in that arena and understands the risks and benefits of each trial. So we have to ensure also, obviously we want to protect every trial, every research participant, but if there are any adverse events, we have to be prepared to provide care for those if there are any adverse events. And occasionally we will notify primary doctors or other specialists that their patients are in trials. A lot of times the physicians know this, but sometimes it's in our best interest to let the whole care team know that they're part of a trial. If we have people withdraw, which is actually very common, I, as a clinical investigator, always wanted to know why. Was it a problem with the medication? Was it a problem the way we did the study? Could the patient not get to the hospital in a reasonable fashion? There's a variety of reasons why people drop out of trials, and sometimes you can learn about the efficiency and or issues that you're having with your trial staff if you learn why participants are withdrawing. Communication is key and the root of probably most issues that we see in general. So, PIs need to have that responsibility to communicate effectively, read their emails, reply, etc. But really, the IRB, obviously, as you've probably just previously heard from Debbie, you will need to get, as a PI, obviously approval before the study begins, provide the IRB with all the necessary information. And most IRBs will really spell this out well and have a fairly rigid, well thought out algorithm for PIs to follow for submitting information, and that's important. But the PI needs to be aware of all that information and how to submit it and to do it appropriately. So all documents will need to be provided by the PI and the study team for review before you even start the trial. You need to get approval first. So, and that's all about communication. So obviously a protocol will be included with any study, and the IRB obviously will review that and ensure that it's appropriate. And therefore the PI has to follow the letter of the protocol to the T, because you really, deviations, there are a few exceptions, but deviations in general will need agreements and prior approval if they do occur. So unless it's for the safety of the patient. So you're really going to conduct this trial really protocol based. And then if there are any deviations or issues, they have to be documented, explained, and reviewed. So I really think that any untoward changes from protocols, issues with patients, or in general should be communicated immediately with the IRB to see if there are regulatory issues that have to be followed or other patient care situations that may need further input. So I'm a fan of over communicating. So for all PIs, I think keeping in contact with your IRB and everyone on your team almost excessively will do you better in the long run than trying to sit on an island and do it all yourself. So what's important about randomization and unblinding and just a part of the protocol in general, you really obviously, like I mentioned, need to follow the protocols exactly. And there are randomization procedures of how you pick a variety of either patients, medications, etc. This really has to be followed appropriately because in general, when any statistical analysis is done, it's done based on the process that you've used for your randomization. So any change upward in protocol can affect, obviously, downstream your data, and then can affect the outcome, and we don't want that. So we don't really break randomization codes or look under the hood ever unless it's for patient care. And for most protocols, we'll have mechanisms for this if you need that. If there is any unblinding or any difficulty, like we said, notify the sponsor and the IRB. I think that's the simplest thing to do. And most sponsors have phone numbers. They 24/7 can answer the phone, help you with any processes, and they're really pretty good about these things. But notifying the IRB as well, because you may have to stop the trial for a little bit or reevaluate the way you're doing things if you're having issues. Good clinical practice is important for informed consent. And I think, as was mentioned before, you have to have the ethical principles that really avoid emotional language. Patients have the right to refuse. They have the right to participate. They shouldn't be forced one way or the other. And they have all, there should be no language in there to have any have them waive their legal rights. So this is really important that that is respect of the individual. And obviously the Declaration of Helsinki and good clinical practice really dictates that patient comes first, and they have every right to drop out at any time or ask any questions. I think that's really critical. If there are any new information or consent documents or any documents require any updating, that will have to be communicated with the IRB, and obviously the sponsor will be involved as well. So we really want to respect the rights of every individual to be a participant, and they have the right to the best possible care and protection that you can provide for them. And a lot of patients don't completely understand the language, so you have to be there to appropriately explain it to them. It's not rushing through, forcing, or pushing towards one way or the other. It's their option, and as a PI, you want everyone to enroll, but it has to be done safely and correctly. One, legally, that's inappropriate if you use such language, but two, patients, have the right, especially if they can't completely understand, to get that information so they understand what decisions they're making for themselves, their bodies, their family members, etc. Progress reports, as all reports, have to be done in a timely fashion. In general, there's a written report at least annually, sometimes more, depending on IRB and sponsors, etc. That's important. And as mentioned, if there are any changes anywhere to any language, the protocol, risk, etc., that has to be documented and updated and discussed. And that's your communication with the IRB and the sponsor. That's really important. I mean, this stuff isn't difficult, but what we see a lot of principal investigators is sort of neglectful of timelines and maybe not as efficient. But then that, as an IRB myself, would assume that they're not taking an active interest or something negative is happening. So that's why I'm such an excessive communicator. So safety is key, and like we said, our job as PIs and the IRB is to protect the patients and hopefully get the cleanest data in the most appropriate fashion. So any adverse event, especially serious, obviously fast, you have to immediately report them. All serious adverse events, and then follow up with a written report. Now, there's some protocols that don't require immediate reporting, so just know your protocol really well about the timeline. But in general, any serious adverse event really needs to be reported immediately. We avoid personal identifiers or any information on a study sheet or data collection that can identify an individual. In general, we'll use codes, numbers, etc., to separate themselves and protect the confidentiality of the patient. That's our promise to the patients as well. And then complying with all regulatory requirements of reporting. There are various mechanisms to report and timelines. So serious adverse reactions, in general, will need regulatory paperwork and IRB paperwork. If in doubt, just ask your IRB. That's really important. Or obviously become intimately familiar with your protocol, which you will, and with all documents. So you should have an idea of the mechanism and the timeline for reporting adverse events. Next, please. So we need to be careful of a variety of these things, and we've seen over the years all of these. I just wanted to point them out, so hopefully it can protect you guys if you are principal investigators, or for some oversight if you're on IRB. But the key first bullet point of what I mentioned is that as the investigator, they have the ultimate responsibility to understanding the protocol and having complete oversight. And that's important. And then you can have delegation of responsibility with co-investigators and a variety of research staff, but that has to be documented, and everyone should understand what those responsibilities are, as opposed to, I thought they knew what they were doing, or I told them what to do. I think more documentation is better as well. All protocols have to be adhered to by the letter. Any deviation can alter everything from statistical data to patient effects. So we have to be very careful to adhere exactly to requirements. Source documents have to be fully vetted, completely organized and up to date, and that's important as well. Any changes or any issues have to be reported to the IRB, of course, and of course done in an appropriate fashion. Consents have to be updated and signed. I've seen a lot where they forget to get them signed, or they store them and forget where they keep them. These things are critically important, that they are protected and that you as the PI know where they are. And then all other records from accountability, et cetera, especially when you're dealing with sponsors and medications and any drugs, you have to have rigorous appropriate accountability records and documentation of that. Otherwise, you will be in non-compliance and could create issues down the line. Obviously, we mentioned these things can be simple mistakes, but it's still your responsibility. So even those simple mistakes can really be costly down the line. The consent is a big one. Know where they are, keep it handy, store it appropriately, et cetera. Any changes have to be communicated as well. Specimen handling and processing labels can get mishandled, specimens can be misplaced. That has to be really, really rigidly followed and adhered to, because we just see problems from the beginning of staff that just don't totally understand where things go or what to do with the specimens. So you as a PI should be very clear about where things stand and how things are handled. But the consequences of any of these simple mistakes, all the way up to large mistakes, could be, simply just data is going to be compromised. It could be lost. You could have a variety of issues, and then the whole study becomes invalid. But then investigations evaluations from other authorities, such as the federal government, or even your own institution, could create a risk for you as a PI. You can be banned from any studies whatsoever. It can lead to fines. It can lead to institutional penalties. So you can put your career at risk by not paying attention to the details. Most minor issues are excusable and can be corrected and documented as long as you recognize them and you're honest and upfront. But if large issues occur, we've seen healthcare systems in the news for these issues with large fines and damage to their reputation, with principal investigators being named. So you have to be real careful. So you really need to just understand your responsibilities. My job here is not to scare you. I think it's an honor to be a principal investigator. I think it's wonderful to work with all learners and researchers, et cetera, and can really get some critical information that can help patients down the line. But you just have to understand what you're getting involved with. You as a PI and your staff need to know all the regulations and the responsibilities, understand the protocol, read it. And, at first it's long and tedious, your first couple of trials, but then there's a lot of repetition, and it actually becomes fun to do research, and you really become, these protocols well enough, you can almost rehearse them, but you really should be very comfortable with inclusion and exclusion criteria, every test and observation that will be made, and what documentation is really. So the take-home message is the principal investigator is responsible for really making sure that everything is done by protocol, good clinical practice, regulations, et cetera. Documentation must be there, signed and dated is really critical. We've seen lots of paperwork turned in where people forget to sign it. It'll be returned if not, but that's really important. And then you're ensuring that during the trial that everything that's being done there is done accurately, completely, and should be verifiable, because that's really your responsibility. So obviously, good clinical practice is understanding patient safety. Patient comes first, regardless of who, what, where, when, always. And the integrity of the data comes second, but they're both critical. Monitoring, continually quality management will help ensure compliance. And like I mentioned, everything really falls on the principal investigator.
Okay. All right. So we're going to move on. Again, we have a lot here, so I'm going to go through some of this, skip through some of this. I want to ask this general question: anyone involved in any oncology trials? Okay, so I can skip through a lot of trials because a lot of these trial designs are exclusive to oncology. They tend to be the ones to come up with the most creative and complex designs. So they'll be in the slide deck for you as reference, but I'm not going to spend time talking about those. So deciding, how to design your clinical trial. I mean, the gold standard in clinical research is the randomized control trial, right? That's the most significant trial design that removes investigator bias. Human bias is extraordinarily impactful on studies, and there is no intent. It's not a malicious intent. We don't intend to be biased, but we just are, and it's almost impossible to remove that. So how do you do it? Well, you randomize, you control, and you blind it. And that's really the way to remove that. So because of that, statistically, scientifically speaking, that tends to be seen as the most substantial trial design. So subjects are randomly assigned to treatment groups or arms, or to a control, whether it's placebo. Randomization prevents participants learning what group they were assigned to. So it also prevents them from being biased, right? So if they know what they always want to know, they're always going to want to know if they got the study drug or if they got the study treatment. But as soon as they know, their responses then are biased. The placebo effect, right? Some advantage. So each of these trial designs, I just have some advantages, some disadvantages, and then some human subjects implications just to consider when you're deciding for your own investigator-initiated type projects. The advantages not only consider the gold standard, but they're non-observational, based purely on objective data. So the outcome is going to be whatever the outcome is, right? You can trust that data. It balances the participant characteristics and removes researcher bias. Some of the drawbacks, though, obviously, these are very expensive. I mean, pharmaceutical industry trials now, I think the recent estimate is that they cost about $1 million a day, which in part is why, drug prices are so high. It's not justified, but that's really the reason why. You may have a trial that even, goes through a year or so and then stops. And how many drugs don't actually ever even get through, phase 2 to get to approval? These things are extraordinarily costly. The effort in order to be able to effectively run a randomized controlled trial, it can't just be the investigator. Like if it's just you, you're not going to be able to do that. You're going to have a team, right? A team of support research coordinators and other support group mechanisms to be able to manage that type of trial. There are often in some of the literature considered problems with generalizability. So I don't know that this is exclusive to a randomized controlled trial, but your cohort is based on the volunteer group, right? So in some conditions, are there certain groups that are more likely to volunteer versus other groups? And if so, then that already is going to kind of conflate the result a bit, right? So it's sometimes seen as a drawback of randomized controlled trial, but again, I think that applies to a lot of those study designs. Your groups are based on who's willing to volunteer. Some human subjects considerations. It's very important to clearly explain, and we see a lot of consent forms that don't do this adequately, that you have a one-on-one chance of receiving the study of medication or a 2-in-1 chance. Sometimes they'll reference, like flipping a coin. There is a chance that you will receive placebo, which is this. It is not an active study agent. And then the consent also needs to describe if there is an opportunity for like a crossover study design, where for a certain period of time, you may be able to cross over to the active agent. Internal and external controls are really essential in these because there tend to be blinding mechanisms. There need to be some internal and external controls. Internal will be like your coordinating center, or your CRO, the contract research organization that are doing, like Debbie mentioned, the monitoring, right? There's interim monitoring visits. And then the external control would be like the IRB and the Data Safety Monitoring Board. I think it's really important if you're an institution that is moving into randomized control trials, that your IRB or Human Subjects Research Protection Program has a mechanism in place to do some internal monitoring. We have our own compliance administrator who will go out and visit these trials, and sometimes the coordinator will say, well, the monitor just came and visited, the sponsored monitor. Study sponsors are interested in getting that product to market. That is their objective. They're working for a commercial industry. They are not necessarily there for exclusively looking at human subjects protections. We are. The internal monitoring is looking at very different things than that external monitor is. And oftentimes they outsource their monitor activities to contract research organizations. Sometimes they don't have, many years of experience, sometimes only months. So the uncontrolled trial, where the efficacy of a treatment, it's compared to a group of patients without a control group. There is a control. Oftentimes they'll pull from like medical record data, right, of existing treatment and compare. This is more typical in early phase trials to investigate like dosing levels and particular characteristics of a new drug. The advantage is obviously there's less patient burden in this because you don't have that control group to enroll. And oftentimes it's a little less complex for a study team to understand and manage. And there are more flexibilities, obviously, in the overall design of the protocols. The drawbacks, obviously, it's going to introduce bias. You can't help that. You can't remove that. If you know what that patient is being treated with, it's difficult then to even remove that investigator's bias. And then often there are problems with the generalizability based on the cohort of participants who volunteered, same as the randomized control trial. Human subjects, if it's an early phase trial, I think it's really important to emphasize that. It's not uncommon for consent forms to maybe try and downplay certain things where if risks are not yet known, that actually is more risky than if you have like pages and pages of risk, right? Because if you have pages of risk, that means that an earlier trial identified those risks. If you are introducing an active agent that may not have been administered in humans before, you don't know what those risks are yet. You can make assumptions, but you don't know. But as these early reports come in, that's how that risk, that list of risks is identified. So you don't have any because it's an early phase trial, and it's really important to emphasize what that means to that participant. There may be less existing data to demonstrate effectiveness. Uncontrolled trials may not lead to FDA approval. So obviously early phase trials aren't going to lead to approval right away. They're going to have to go through other phases to get there. I usually bring up, is it ethical to use existing medical record data to sort of stand in place as that surrogate control group? Because that medical information was obtained for clinical purposes, right? It is allowable under the regulations to then mine that data, to go in that data, to do like chart reviews for research purposes. Then the question is that ethical? Well, it's ethical if your privacy statement that patients sign when they come in the door is very clear. And every time if I work at a new institution, the first thing I ask for is their privacy statement, because I want to know under what terms is that medical information collected. If it prohibits the use for research purposes, even if it's allowable under those regulations, it's really not ethical to be able to do that. But most privacy statements today leave a provision for we can pretty much do anything with your data that is collected. But it's a good idea to check. So non-randomized is like randomized versus non-randomizing. The investigator controls the allocation. Obviously, a disadvantage is going to be bias, right? But some advantages are going to be, they can tend to be quicker, put them together quicker. They're easier to design, easier to write, easier to follow, less patient burden. It may be able, to allow for the research in areas where like a randomized controlled trial is really not possible. If you're let's say looking at things like effects of like a smoking ban or something, it's dealing with like abortion or smoking, things where it would really be unethical to have a control group, right? So there may be cases where not randomizing would be is essential, is needed. I'm sorry, these first two should be in the advantages, right? It's lower cost and shorter time. Those are really advantages of a non-randomized trial. Some human subjects considerations. Some feel it's ethically, well, it is. It's always ethically irresponsible to study drug effectiveness in a non-randomized trial. Anything that you can't remove that investigator bias, you don't want to try and investigate like the safety and effectiveness of a study treatment, any study treatment really. Some consider the reduction of the cost and time should be worth the subject burden and safety in favor of a randomized trial. I should have mentioned at the beginning, all these trial designs, they're not mutually exclusive. So you can have many different designs in one protocol, right? Randomized, controlled, blinded, so on and so forth. So there may be multiple layers of these specific trial designs in any given protocol. So open label versus blind. An open label study is a trial in which investigators are aware of what group the patients were allocated to. The use of subjective variables by the investigators may involuntarily condition the results based on the information that the investigator has regarding that patient's treatment. So advantages, this is oftentimes a fairly simple study design, easy to manage, easy for the study team to follow, and easy for subjects to understand. But the drawbacks, again, you see this bias continuously coming up in anything that's not a randomized controlled trial. Potential for conflict of interest. I see, you know, sometimes open label are really more marketing protocols, right? Where they may have already, it may be an approved drug and they're doing like an additional, sometimes you'll see like phase 4 trials. I'm very hesitant. I'm very suspicious of phase 4 trials because oftentimes these are marketing protocols for the industry. They're really not to do anything to benefit patients directly. So then is it allowable or isn't it? Well, at the end of the day, I think it's, you know, if the institution is okay with it, then it's up to the participant to decide. But then they need to know, you know, I will push back sometimes on the consent forms to say, no, this is a marketing trial. You need to say that your objectives here are really to do, you know, marketing research and just be clear about it. And then it's up to the patient to decide whether or not they want to participate in that. It may be beneficial to them, right? They get the study medication paid for by the sponsor as opposed to insurance. Blinding, I think we know what blinding is. Reducing this risk of bias, right? Where you blind it. You can have either a single blind or double blind, or even in some cases triple blind, or even the statisticians don't even know what the allocation was. And Brian mentioned, you know, unblinding. So that's really one of the potential, you know, human subjects considerations with blinded versus unblinded trials. There tends to be a reluctance to unblind, right? If it's like a patient safety issue, that should never be a consideration. They're going to, you know, it's going to confound our data. Well, if it's a patient safety issue, that always is primary. That's always number one. Some advantages of blind studies carries the highest level of scientific validity. So you'll have a randomized control double blind study, right? So that oftentimes that blinding will be worked into that randomized control trial. If the results show efficacy and safety, particularly in like a phase 2 or 3, there's a higher chance of additional studies leading to market approval. And I don't want to dump on like, you know, industry sponsors that they're all after the getting that product to market. Of course we want that, right? If it's showing efficacy and safety and it's going to be beneficial to a population, we really want to get that to market. Oftentimes in late phase trials, I'll ask, you know, is this going to continue? Like at this point, there is a lot of evidence to suggest that this is beneficial, right? Is this going to be possible if it's like a long-term drug, blood pressure medication or diabetes or something like that? Are they going to be able to continue to have access to it? You know, if it doesn't get marketed, then they won't. So that's always important to consider. Some disadvantages, they're very complex. When you see those long trial names, you know, where the titles go on forever, the more types they add, the more complex it is. And the more complex it is, the more people you need running it. And more, you know, manuals in place, right, to control these things. There's no room for, I've already mentioned that, there's no flexibility in running research. Those protocols are very well defined. If you're doing like an investigator initiated, so let's say you're doing like a grant funding where you're expected to write that protocol yourself, you know, from an IRB perspective, I'll scrutinize those and I'll ask for more and more and more detail. And, you know, it annoys the investigators. We get that. But I want it to be impenetrable, right? That there's no flexibility. And you want that too, because then your data results are going to be more solid, more suitable for publication and more suitable for future studies. The worst thing you read about in like research misconduct is when there are studies, you know, like data fabrication or things like that, right? And then those studies have continued on to other studies that have relied on that data to support them, right? It compounds that problem. So for the most part, what we've seen is these are all coming from industry sponsors. So the randomization oftentimes be done at the sponsor level. If you're designing these in-house, it's more like an in-house, and you want to, you know, design a blinded study, there's going to have to be some kind of like honest broker. There's going to have to be sort of this third party, this data coordinating center, and there's going to have to be a randomization mechanism, a mechanism by which they're randomized that's not investigator controlled, that is apart and outside, exclusive from the study team and the study protocol. But it does become complex. I mean, we do have cases. know of any examples off the top of my head of internal, like investigator initiated type trials. I know that they've done them blinded, but I'm not aware how they, what their process was for doing that. It needs to involve either some kind of software or some other party involved to be a part of that randomization process and to protect that blind. So there's someone protecting that blind, right? There's someone on the other side of that curtain that's assisting to manage that, and they're not part of the study team. So I have a few other trial designs, and then I also had some slides about the those human subjects clinical trial forms, those NIH forms. I don't prefer to go maybe in that direction. All right, so I'm going to skip ahead. So these forms are, they are unnecessarily complex, right? Because it's NIH, and they really are, and it's very confusing. I think the first thing to do if you're required to being asked to complete these forms is download the supplemental instruction manual. It's like 180 pages. It's extremely dense, but that's your guide. And it gives you step-by-step instructions on how to fill out every section of these forms. And they will return it if anything is wrong, right? If you're not following the specified formats, some of the attachments, like some attachments that go along with some of these questions, some of them, there are specific format requirements. Others, it just says there's no page limit. That's why you want to read that manual for every question that you get to. So before you get to the human subjects and clinical trial information form, there's some information that you will have already populated in what's called the other project information form. Make sure you're doing that first. Like, don't jump to this HSCT form. Make sure you're completing the whole, it's that R&R, the standard R&R application form, right? And one of those sections is the other project information form, because it'll ask some human subjects information questions, and the responses to those questions will pre-populate in the human subjects clinical trial form. One of the initial questions is, does this project, are human subjects involved? If you answer yes, is the use of human specimens or data involved? So you may include human specimens or data, but it may not be considered human subjects research. That is extraordinarily confusing to a lot of people. So under the federal regulations, if you're receiving specimens or data from an outside source and you do not have access to any of the identifiers, and the collection protocol or your protocol defines that you will never be given that, whoever collected those samples or data will never share those identifiers with you. If those are true, then you're not engaged in human subjects research. So you would say, yes, human subjects are involved, but no, it's not human subjects research. And then there's a section for you to provide a justification for that. Let me skip ahead a little bit here. So here's a link to the NIH policy on human subjects research. They have a lot of tools. They have like a lot of decision tools. They have like questionnaires that you can answer questions. And as you go through the questions, it'll sort of spit out what direction you should be going. So here, this third paragraph, if you check node, are human subjects involved, but your application proposes using specimens or data, that's when you'll need to provide justification for, I'm getting specimens and data. So I'm getting tissue samples, but they're surgical discard and I have no identifiers associated and I'll never have access to them. That's then what your justification would say. Because then hopefully the NIH reviewer will say, oh, right, human subjects aren't involved. So that's sometimes hard to grapple with. So the first question we always ask is it research? Well, we know it's research because these funding mechanisms only fund research. And then the second question we ask is it human subjects? So that's when the data specimens question comes in, right? Because there are certain research activities involving these specimens or human data that's not human subjects. And then the third question we ask is, do you qualify for an exemption? This is when I would consult with an IRB. I don't like it that the NIH almost expects the investigators to know what exemption category they're going to fall into. These are extraordinarily narrowly defined in the regulations, and you really need that IRB consultation to know whether or not that your proposed project is going to fall into an exempt category. Here's some graphics that are on that NIH Human Subjects Help page. to help answer those questions about whether or not it's exempt. I'm going to skip ahead in some of these. So this is that other project information form, the last question. So they ask about the exemptions, and then last question is the IRB approval pending? If it's not yet approved, then it's pending, and then leave the rest of it blank. So in that instruction sheet, it'll tell you, don't respond here if you answered no here. That always makes folks nervous, right? Submit a blank space to the NIH. But that instruction manual will tell you that. So then after you finish that other project information form, and then the rest of your application, then when you get to the human subjects and clinical trials information form, some of that information will already be populated. Maybe a closing remark, and maybe you have a couple minutes for questions. Each one of these sections here, these study records, you could have multiple study records for one proposal. So you could be submitting a proposal for funding that could include more than one research protocol. If you understand that, then when you go through these forms and read that instruction manual, that'll make more sense, because in a lot of cases, you won't have multiple study records, right? You're going to have one protocol. But when you read through the instructions, it is very confusing for folks. They're like, well, why do I have to submit up to 50 of these? It's just saying that could be a possibility under a particular funding mechanism. I'm going to stop there. It's like one minute. All right. Well, thank you all. I've been very attentive. I know this has been a long chat, but I appreciate your attention. I appreciate y'all being here and inviting me. And so the first slide and last slide, I have my contact information. Feel free to contact me anytime as you're working through either the forms or your protocols, and I'll either answer or I'll put you in contact with someone who can. Thank you.